Okay, ladies and gentlemen, you are welcome. So today we are going to discuss on the general concept of metabolism. Okay, ladies and gentlemen, you are welcome. So I hope you can hear me. So in today's video, we are going to look at the general concept of metabolism. General concept of metabolism. So we are going to start a general revision from beginning of the course. I hope you are all with me. So what is metabolism? Metabolism, ladies and gentlemen, is a sum total of all chemical reactions that are taking place in our body. So when we sum all the reaction, when we sum all the sum of all reactions in a living organism, in a living organism, living organisms, that is what we call metabolism. So the sum total of all biochemical reactions that are taking place in a body of a living organism is what we call metabolism. And we have different types of metabolism. So ladies and gentlemen, what are those different types of metabolism? Can you write them for us? The different types of metabolism. Who can tell us number one, the different types of metabolism? Okay, so number one, we have what you call catabolism. We have catabolism number one. And then apart from catabolism, then we also have number two, we have anabolism. We have anabolism. So as I said, we are going to start from the beginning of this course for this our live discussion. So now, what is catabolism? So when you say catabolism is when you have any reaction where a complex molecules break down to produce smaller ones and generate energy in the process is what you call catabolism. Let's say now you have a larger molecules like this. Let's say this molecules is carbohydrate, carbohydrates. So when you break these larger molecules into smaller molecules like this, so the breaking down of complex molecules into smaller one, and in the process, there is a release of energy. That is, energy is released. This is what you call catabolism. So this is catabolism. So in catabolism is when you have a larger molecules break down into smaller one and generate energy in the process. That is what you call catabolism. Then apart from catabolism, ladies and gentlemen, we are also going to explain the concept of anabolism. So what is anabolism? Anabolism is when you have a smaller molecules. When you have a smaller molecules like this, joined together to produce a larger one like this, and in the process, energy, you see here, energy is released. So in the process of anabolism, energy is added. Energy is added. So as we can see, in the process, you have a smaller molecules. So when you have a smaller molecules joined together to produce a bigger or a larger molecules, you see here, you have a carbohydrate as a larger molecules break down to produce smaller one and release energy, that is catabolism. And then when you have a smaller molecules joined together to produce a larger molecules and in the process energy is added, that is what you call anabolism. And then apart from catabolism, when you check other textbooks, you will also see what you call amphibolism. That is number 3. We have amphibolism. So what is amphibolism? In the case of amphibolism, is when you have catabolism and anabolism taking place at the same time. That is what we describe as amphibolism. So amphibolism is when catabolism and anabolism are taking place at the same time in a cell. That is what we describe as anabolism. So without wasting much of our time, I told you that we are going to have a general discussion on glycolysis and apart from glycolysis, and we are also going to look at Krebs cycle. So now, ladies and gentlemen, let's start with the basic concept of glycolysis. Glycolysis. So in glyco means carbohydrate or sugar. So in the process of glycolysis, you have glucose. So a glycolysis is a series of enzymatic reaction, where a glucose molecules break down to produce two molecules of pyruvate, two molecules of ATP, and two molecules of NADPH. So in the process of glycolysis, glucose, because the word lysis means breaking down, the word lysis means breaking down. So when you have glucose break down to produce two molecules of pyruvate plus two molecules of ATP plus two molecules of NADH. This is what we describe as glycolysis. Okay, good afternoon. So this is what we describe as glycolysis. So at the end of glycolysis, you should put this at the back of your mind, ladies and gentlemen. At the end of glycolysis, you are expecting to have two molecules of pyruvate, two molecules of ATP, and two molecules of NADH plus two molecules of NADH. So at the end of glycolysis, you are expecting to have two molecules of pyruvate, two molecules of ATP, and two molecules of NADH. So glycolysis, please, you shouldn't forget about this. It's simple. It's a process in which glucose is break down to produce two molecules of pyruvate, two molecules of ATP, and two molecules of NADH. But now the question is, where is this glycolysis take place? Where glycolysis take place in a cell, ladies and gentlemen? Please answer this on the comment section of this video. Where glycolysis take place in a cell? Where glycolysis take place in a cell? Answer this, please. Answer it on the comment section. Please answer this on the comment section of this video. Where glycolysis take place in a cell? You don't know? So let me answer it. So glycolysis take place in the cytoplasm. In the cytoplasm. So glycolysis takes place in a cytoplasm of a cell. Remember, you did biochem 241, and we have different organelles of the cells, and most of these metabolic processes are taking place at different organelles of the cell. So therefore, glycolysis takes place in the cytoplasm of a cell. So let me clean this site so that at least we can properly look at the glycolytic pathway. So what is the first reaction in glycolysis? So the first reaction in glycolysis is when you have glucose molecules plus ATP. So glucose plus ATP in the presence of enzymes called hexokinase. Hexokinase. So what this hexokinase do? Remember here is ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. So the word hexokinase, sometimes you cannot actually be able to remember some of this information. That is why you need to understand the function of each and every enzyme. So the word hexokinase, kinase, it is an enzyme that involves a transfer of group. So it is a transferase enzyme that remove one group from one molecules and transfer it to another molecules. Like for example, hexokinase here, remove one phosphate, it remove one phosphate from ATP and add it to glucose 6. So therefore, we are now going to have glucose 6 phosphates. Glucose 6 phosphates plus ADP plus ADP. So as you can see here, you have glucose plus ATP. So one phosphate group from ATP here is transferred to glucose. That is why we have glucose 6 phosphate plus ADP. So ADP means adenine dinucleotides, adenine triphosphate. So this is what happened at this stage. This is the first reaction of glycolysis. So the next thing after this is the conversion of glucose 6 phosphate to fructose 6 phosphate. So therefore, another enzyme is called phosphofructoisomerase. Phosphofructoisomerase. So phosphofructoisomerase is an enzyme that convert glucose 6 phosphate to what place? Let's be following, please. So this enzyme glucose, so the phosphofructoisomerase convert glucose 6 phosphate to which product? What is the product of this reaction? What is the product of this reaction? Glucose 6 phosphate is converted to which product using phosphofructoisomerase? Okay, write it please on the comment section of the video. So what this enzyme does, it converts glucose 6 phosphate to fructose 6 phosphate. It converts it to fructose 6 phosphates. Glycolysis takes place in the cytoplasm of the cell. So this is how the reaction takes place. So the first reaction, glucose plus ATP to produce glucose 6 phosphate, and then the glucose 6 phosphate will be converted into fructose 6 phosphate. But ladies and gentlemen, what is the importance of the first reaction of glycolysis? That is the conversion of glucose to glucose 6 phosphate. So remember, ladies and gentlemen, if you could remember, in the transport system, I told you that glucose and sodium transport into the cell at the same time. That is simple transport. So when glucose gets itself into the cytoplasm of the cell, there is a likelihood that that glucose can easily sneak out from the cell. So we need something that will retain the glucose in the cytoplasm of the cell, that is inside the cell. So that is why we need to phosphorylate it. So the phosphorylation or the glucose 6-phosphate cannot, because of the addition of this hydrophilic phosphate group or this negative charge, that is the phosphate, it cannot sneak out from the cell. So therefore, it will remain there. So this is one of the important reaction of hexokinase. That is the first reaction of glycolysis. Please, is that clear and are you following? Is that clear? Are you following me? Do you now understand the importance of phosphorylation of glucose into glucose 6-phosphate in a cell? Do you understand? Please write it on the comment section of the video. Please, I'm waiting for your comments so that I will understand that you are really following the class. Okay, someone said no. What I said, did you understand the importance of the phosphorylation of glucose in a cell? Let's say this is a cell. Let's say this is a cell. So remember, after you digest carbohydrates, the end product of digestion of carbohydrate is glucose. So let's say this is a glucose in the blood. You have a glucose here in the blood. You have the glucose in the blood. So, but now the question is, how does glucose get itself into the cell? It is using a simple transport with various glucose transporter, although it depends on the types of the cell. So therefore, remember in the simple transport, glucose and sodium ion get themselves into the cell in the same direction and at the same time. So now assuming now you have the glucose inside the cell. So at the same time, these glucose molecules can sneak out or can leave the cell. So how do you prevent, how can you restrict the movement of glucose inside the cell? Is through adding phosphate group. So you add phosphate group using ATP. So that is the importance of this reaction, where you have hexokinase transfer one phosphate group from ATP to to glucose at carbon 6. So that is why we have glucose 6-phosphate, indicating that the phosphate group is attached to carbon 6 of the glucose molecule. I said the importance of the first reaction of glycolysis is when you have your glucose into the cell, you add a phosphate group so that to restrict or to protect or to prevent the glucose molecules from going out of the cell. The phosphate group is a negative charge, and once it's added to the glucose, then the entire molecules become charged. Therefore, the glucose molecules cannot go out of the cell. So that is the importance of the hexokinase, is to add the phosphate group so that to make the glucose 6-phosphate negative in the sense that it cannot go out of the cell. And then the next reaction is the conversion of the glucose 6-phosphate to fructose 6-phosphate using phosphofructoisomerase enzyme. That is the name of the enzymes that do the conversion. Then the next thing is the conversion of glucose 6-phosphate to glucose 1,6-bisphosphate. So you have an enzyme called phosphofructokinase. So phosphofructokinase, it is the third enzymes in glycolysis, where you see from the word here, that is why I told you kinase. Whenever you see kinase, it means it is a transferase enzyme. So kinase here also transfer phosphate group from another molecules of ATP. So therefore, at this point, you are reacting phosphofructose 6-phosphate with another molecule of ATP. So you see, when this phosphofructokinase act on fructose 6-phosphate, we are now going to have fructose 1,6-bisphosphate. What does that mean? You add kinase group, you have kinase enzymes, which will remove one phosphate group from ATP and add it at carbon 1 of the fructose to produce fructose 1,6-bisphosphate plus ADP plus ADP molecules. So here we have ADP and we have fructose 1,6-bisphosphates. Do you understand? So the next thing is the work of another enzyme called aldolase. Aldolase. Another enzyme is called aldolase. What this aldolase did, it's cleave. It will divide the fructose 1,6-bisphosphates into two molecules. So therefore here, we are now going to have two molecules. And these two molecules, when it cleaves, it's going to give us two molecules. So the first one is glycerol dehyde 3-phosphates. Then the other one is dihydroxy acetone phosphates. I hope you are following. So you see, after the formation of fructose 1,6-bisphosphates, the fructose 1,6-bisphosphate will be cleaved or will be divided into two to give dihydroxyacetone phosphates and glyceraldehyde 3-phosphate dehydrogenase. Do you understand? But very importantly, ladies and gentlemen, at this point, dihydroxyacetone phosphates cannot be continue with the glycolysis. So it is not a substrate for glycolysis. It is not a substrate for glycolysis. So therefore, this dihydroxyacetone phosphate must be converted into another molecules of glyceraldehyde 3-phosphate. So therefore, at this point, we are going to have another enzyme called triose phosphate isomerase. Triose phosphates. Triose phosphate isomerase enzyme. So that is the name of the enzymes that combat this hydroxyacetone. Already remember, we have 1 molecules of glyceraldehyde 3-phosphates. So then the next thing, this dihydroxyacetone phosphate will now convert will now be converted into another molecules of glyceraldehyde 3-phosphate. So remember, we already have 1 molecules of glyceraldehyde 3-phosphate. So therefore, by converting this dihydroxyacetone phosphate to another molecules of glyceraldehyde 3-phosphate, it means now we have 2 molecules of glyceraldehyde 3-phosphates dehydrogenase. So we have 2 molecules here. It's not a slide, rather it is a bot. I said we have dihydroxyacetone phosphates will be converted into another molecules of glyceraldehyde 3-phosphate dehydrogenase because dihydroxyacetone phosphate cannot continue glycolysis. So therefore, no glycolysis here. When I said no glycolysis, it means that the dihydroxyacetone phosphate cannot undergo or cannot continue with the glycolysis. So therefore, it must be converted into another molecules of glyceraldehyde 3-phosphate. So that is why here at the end, we now have 2 molecules of glyceraldehyde 3-phosphates. So then what happens next? The next reaction is the conversion of these 2 molecules of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate using an enzyme called glyceraldehyde 3-phosphate 3-phosphate dehydrogenase enzyme. Dehydrogenase. So that is the name of the enzyme. So we have glyceraldehyde 3-phosphate dehydrogenase. So what this enzyme does, it will now convert this glyceraldehyde 3-phosphate dehydrogenase into 1,3-bisphosphoglycerates. And at this point, these enzymes require NAD+. It requires NAD+, which will be converted into NADH plus H. So what I want you to understand, ladies and gentlemen, since this is 2 molecules, so here is also going to be 2. So therefore, everything now is going to be 2 here. So therefore, at this point, we have 2 molecules of NADPH. It is only the enzyme that will remain as 1 molecule, 2 as 1. But the glyceraldehyde 3-phosphate dehydrogenase is already 2. So therefore, the product is also going to be 2. Please, ladies and gentlemen, are you following the class? Are you following? Are you following before you continue? Okay, thank you, sir, dear. So now, let me continue. I will just clean this. So you see, what we have here is we have 1,3-bisphosphoglycerate. So this 1,3-bisphosphoglycerate is going to be converted into 3-phosphoglycerates. 3-phosphoglycerates. And the enzymes that do this work is phosphoglycerate, phosphoglyceralkinase or glyceral kinase, phosphoglyceral or glyceral kinase. So this enzyme, what it does, we have ADP here, and then one phosphate, the phosphate here at position 1 will be added to ADP to produce ATP. So ladies and gentlemen, please look at this reaction. Sabat, can you move gradually, sir? I'm a little bit confused from aldolase, okay? We have fructose 1,6-bisphosphates. And then the fructose 1,6-bisphosphates will now be cleaved into two using the aldolase enzyme. So aldolase enzymes break down fructose 1,6-bisphosphate into two to give dihydroxyacetone phosphates and glyceraldehyde 3-phosphates. But very unfortunate, dihydroxyacetone phosphate cannot continue with glycolysis. So it cannot continue with glycolysis. Therefore, it will now be converted into a substrate that can enter and continue glycolysis. So that is why it converts itself from the dihydroxyacetone to glyceraldehyde 3-phosphates. So already we have 1 molecule of glyceraldehyde 3-phosphates. So when dihydroxyacetone phosphate is converted into another molecule of dihydroxyacetone phosphate, that means we are going to have 2 molecules of glyceraldehyde 3-phosphates. And then the enzymes that convert the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate is triose phosphate isomerase or triphosphate isomerase. So now the glyceraldehyde 3-phosphate dehydrogenase will now be converted into 1,3-phosphoglycerate using an enzyme called glyceraldehyde 3-phosphate dehydrogenase. So that is the name of the enzyme that converts the glyceraldehyde 3-phosphates to 1,3-phosphoglycerates. Sorry, bisphosphoglycerates. And then the enzymes that did this work is glyceraldehyde 3-phosphate dehydrogenase. So then the next thing, and everything now is now going to be two molecules. And this glyceraldehyde 3-phosphate dehydrogenase, it requires NADH as a coenzyme. So that is why NAD plus will enter, and then you generate NADH. So because here we are talking about 2 molecules, and then the product is also 2 molecules. So the NADH is also going to be 2 molecules. So then the next thing is 1,3-bisphosphoglycerate will be converted into 3-phosphoglycerate. And the enzymes that convert the 1,3-bisphosphoglycerate to 3-phosphoglycerate is phosphoglycerol kinase. That is the name of the enzyme. And how these enzymes work. These enzymes will remove the phosphate group at carbon 1 and add it to ADP. It will now add it to ADP to generate ATP. And don't forget, ladies and gentlemen, here we are talking about 2 molecules. So therefore, here we are also the 3-phosphoglycerate is going to be 2. Is also going to be 2. So therefore, here we have 2. And then we have 2 molecules of NADH and then 2 molecules of ATP. So see from here, ATP is generated. And how many ATP we generate? 2 molecules of ATP at this stage. And this is what we call substrate level phosphorylation. What is substrate level phosphorylation? A substrate level phosphorylation is a situation where ATP is generated from a substrate. The substrate here is 1,3-bisphosphoglycerate, and you remove phosphate here and you add it to ADP, and that is why you have ATP. So therefore, we have 2 molecules of ATP. This is what we call substrate level phosphorylation. And we have 2 ways in which energy are generated in a cell. We have oxidative phosphorylation and we have substrate level phosphorylation. So these are typical examples where ATP are generated from a substrate. Like the substrate here is 1,3-bisphosphoglycerate, and the phosphate is removed from the substrate and added to ADP to generate 2 molecules of ATP. I hope you understand. So then the next thing you see, we have 3 phosphoglycerate. Please bear with me. We have 3 phosphoglycerate. So these 3 phosphoglycerate will be converted into, you see, it's 3 phosphoglycerate and there are 2 molecules. So it will be converted into, sorry, I think I missed something. Is it 1,3? Let me ask you. Is it 1,2-bisphosphoglycerate or 1,3-bisphosphoglycerate? Let me ask you. The product here, is it 1,3-bisphosphoglycerate or 1,2-bisphosphoglycerate? Answer me, please. Please, I'm waiting for you. Please, who can answer this? Is it 1,2-bisphosphoglycerate or 1,3-bisphosphoglycerate? Yes. Alaikum salam. Mala Umar. Ina ɗan class. Zan zo insha Allah. Okay, please answer it. Which one? Who can answer it? Am I correct or we missed something? Excellent. So no, it's 1,3-bisphosphoglycerate. Thank you, sir. I just got connected, sir. Please, can you make it blurry light more visible? Actually, the problem is that, okay, let me try something here. Do you see it more like this? So it's actually 3-phosphoglycerate. So the 3-phosphoglycerate will be converted into 2-phosphoglycerates. It will be converted into 2-phosphoglycerate using phosphoglycerate kinase. So it will be converted into, phosphoglycerate mutase, phosphoglycerate mutase. That is the name of the enzyme. So 3-phosphoglycerate will be converted into 2-phosphoglycerates. 3-phosphoglycerate will be converted into 2 molecules of phosphoglycerates. So then the next thing is what? Is the conversion of these two phosphoglycerates to phosphoenol pyruvate. Phosphoenol, phosphoenol, phosphoenol pyruvates. We have phosphoenol pyruvates. We have phosphoenol pyruvate. And then from phosphoenol pyruvate, the enzymes that catalyze the conversion of these enzymes to this phosphoenol pyruvate is enolase. It's enolase enzyme. So we have two molecules of phosphoenol pyruvate. And then the next thing is the conversion of the phosphoenol pyruvate to pyruvates. 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 And at this point, you have, of course, it's two molecules. So at this point, you see we have phosphoenol pyruvate. So the phosphate from here will be added to ADP to produce ATP. And remember, we are dealing with two molecules. So therefore, here is going to be two molecules, and here is going to be two molecules. So ladies and gentlemen, this is glycolysis. This is a clear description of glycolysis. We started from glucose, and then we end up of two molecules of pyruvates. And don't forget, ladies and gentlemen, I told you that in glycolysis, we have three phases. We have three phases of glycolysis. We have energy generating phase, we have energy generating phase, we have energy generating phase, that is number one. And then we have energy producing phase. Oh, sorry. The first one is not energy generating phase, it's energy investment. Energy investment. Energy investment phase. And then we have splitting phase. So when you check some textbooks, they will tell you that there are three phases. There are three phases. And then number three is energy generating phase. Energy generating phase. So why do you call it energy generating phase and energy investment phase? It is called energy investment phase because at the initial stage of glycolysis, you borrowed 2 ATP because the aim of the glycolysis is to generate ATP and other biomolecules. So, but you are starting with molecules of ATP because in fact, glycolysis is usually taking place in a cell or in a body when there is low glucose. When there is low ATP. So when there is low ATP, usually glycolysis takes place in our body. So now we have one ATP here. We have another molecules of ATP here. So that is why it is called energy investment phase. Because at this phase, you have to borrow ATP from energy, the remaining energy left in the body, and invest it. So therefore, two molecules of ATP are invested. And then the splitting phase is here, where fructose 1,6-bisphosphate is converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphates. That is the splitting phase because you have one molecule divide itself or split itself into two. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphates. That is splitting phase. And then the next one is energy generating phase. So you see, let me tell you, it is very important to understand this glycolysis very well because you must be asked. Krebs cycle and glycolysis usually come in the exam. So please get it very well. So where did we, because sometimes they will ask you state three reactions or two reactions in which energy are generated, or state two reactions or two stages of glycolysis in which energy are invested. So here you can see two molecules of ATP are generated, and then another two molecules of ATP are generated here. So at the end of glycolysis, you produce four molecules of ATP, but the net ATP is going to be two. Why is it that the net energy is going to be two? Because you invested two energy, so you are going to pay back those two ATP, and then you will now left with a net energy of two. So then the next thing is two molecules of pyruvate. These are what you are going to produce. I think if you check, so we generated two ATP, two molecules of pyruvate, and then two molecules of NADH. So ladies and gentlemen, this is where I'm going to stop, and I hope you understand the concept. Please follow me and subscribe to my YouTube channel. See you next.